Welcome and good day to all of you. In this video session we are going to see about an overview of proteomics. Proteomics is the large-scale, systematic study of proteins, particularly their structures and function in a cell, tissue, or organism. The term proteomics was coined in 1994 by Mark Wilkins. Proteomics includes many traditional scientific disciplines including biology, protein biochemistry, analytical chemistry, and other newer disciplines such as bioinformatics and molecular biology. Protein identification and quantification, it helps in identifying the types of proteins present in a biological sample. It helps in measuring their abundance to understand expression levels under different conditions. Functional characterization determines the biological roles and activities of proteins. Helps in understanding how proteins participate in cellular processes and pathways. Structural analysis studies the three-dimensional structure of proteins. Provides insights into protein function, interactions, and mechanism of action. Post-translational modification (PTM) mapping identifies chemical modifications like phosphorylation or glycosylation after protein synthesis. Helps understand regulation, activation, and functional diversity of proteins. Biomarker discovery and diagnostics detects specific protein markers associated with diseases. Aids in early diagnosis, prognosis, and monitoring of treatment response. Drug target identification identifies proteins that can be targeted for therapeutic intervention. Supports drug development by revealing potential targets and mechanisms. Techniques used in proteomics study: 2D gel electrophoresis, ICAT, isotope-coded affinity tags, mass spectroscopy, MALDI, PMF, peptide mass fingerprinting. Structural proteomics, it focuses on determining the 3D structure of proteins and protein complexes. Protein folding, structure, function relationship, X-ray crystallography, NMR spectroscopy. Functional proteomics, it focuses on the studies of protein functions and interactions. It involves protein-protein interactions, enzyme activity studies, signal transduction pathways and protein localization. Clinical proteomics, the application of proteomics in disease diagnosis and treatment with the use of biomarker discovery, cancer proteomics, personalized medicine, drug target identification. Quantitative proteomics, it measures changes in protein expression levels. Post-translational modification, PTM, proteomics, it is a studies in which chemical modifications of proteins after synthesis is done like phosphorylation, glycosylation, acetylation, ubiquitination. Comparative proteomics, it compares protein expression between healthy versus diseased tissues, different developmental stages. Computational proteomics, bioinformatics uses computational tools for protein identification, data analysis, protein structure prediction and network analysis. Plant proteomics, stress response proteins, crop improvement, drought and salinity resistance. Microbial proteomics, pathogen identification, antibiotic resistance studies, host, pathogen interactions. Translational applied proteomics, vaccine development, drug discovery, industrial enzyme production. The proteomics has its application in various research areas like nutrition research, diabetes research, cardiovascular research, autoantibody profiling for the study and treatment of autoimmune disease, urological cancer research, fetal and maternal medicine, neurology, renal disease diagnosis, study of tumor metastasis, and protein biomarker. Thank you for watching. Have a nice day.